Good day. In this lecture, we will be discussing about cell cycle and cancer. So for the outline of this study, in this study, in this lecture, we will be focusing more on the cell cycle, not only on the mitosis. Actually, as I know, you have already learned about mitosis and cell division from way back your elementary and high school days. So this time, we will be focusing on what is the cell cycle. So the mitosis and cell division you have learned in your high school days, in your previous classes, general biology classes. It's just a part of a larger picture, which is the cell cycle. So in this lecture, we will be focusing on the cell cycle and its controls, and we will look slightly just a brief review of the different stages of the M phase, which is the mitosis, and what happens when cell cycle controls has gone haywire, and that is cancer. So the cancer here is not, we will be talking about cancer in the context of cell cycle rather than cancer as a disease itself. Okay, so let's start with the lecture. So the cell cycle has four known phases, and when we say cell cycle, it's basically the life cycle of the cell. Okay, so we have the first phase is the G1 phase. So we start off with G1, and then we have the S phase. It's called the S phase because DNA replication occurs in this phase. And then we have the G2 phase, and then we have your M phase, which is the mitosis and nuclear division. And after the M phase, you get your cytoplasmic division, which is you will end up with two daughter cells. Okay, and then the cell cycle starts again with the G1 phase. So the G1 phase, the S phase, and the G2 phases are called the interphase. So this is what the interface is basically when we look at the microscope. This is the cell, the normal cell that is not going cell division that we see. Although S and G2 phases are the events in the S and G2 phases are quite different from what is happening in the G1 phase. It isn't shown. We don't really see it under the microscope. What is really visible in the movements of the organelles in the cell is when we look at the during mitosis, during cell division. So if we look at this DNA content, so the number of cells versus the relative amount of DNA per cell. So this is just in arbitrary units. So the majority of the cells are in the resting phase in the G1 phase. So cells that are the normal, when if we liken it to our lives, the normal day-to-day life of us is basically we are in a G1 phase. And then less than half will be on the G2 or M phases. The cells in the G2 and M phases, they are the ones that are actively going cell divisions. So if you look at the relative amount of their DNAs, in the G1 phase, you have basically one set of DNA. So we have the 23 pairs. And then when you are in the G2 and M phases, you can see that the number of the DNA per cell is doubled. So instead of 23 pairs, you basically have 46 times 2. So this is 92 chromosomes. Because the reason why there are twice the amount is because in the G2 and the M phases, they are preparing for cell division. So you will end up with two daughter cells that must have 46 chromosomes or 23 pairs of chromosomes. So even on a single cell, in the G2 phase and the M phases, you have twice the amount of DNA. And that also means that what is in between the G1 and the G2 phases is the S phase, and that is where the DNA replication occurs. That is in the S phase. So the cell cycle is a very tightly controlled cycle. So the system that controls it operates much like a timer that triggers the events of the cell cycle in a set sequence. So these sequences are based on connected series of biochemical switches. So how do you control it? We are looking at a very, very complicated and a very, very intricate web of biochemical switches, which initiates specific cell cycle events. So the switches, these are the characteristics of the switches. They are binary. So they are either on or off. There's no in between with them. So it's it's a go or it's a stop. So they are binary and the launch events in a complete irreversible fashion. So that means once it's turned on, you cannot go back. You must go on from that. Is either the cell must undergo apoptosis or must uh, become cancerous in a way. So they must go on forward once they are turned on. You cannot reverse what happened once you have a go on the switch. So it is remarkably robust and reliable because you have backup mechanisms. So if because again, this is very important for the cell, because failure of this one will end up with one of the endpoints of that is tumor growth, which might end up in a cancerous growth, which is basically detrimental for the whole system as a whole, not only to the cell. So basically, you have many backup mechanisms. If one switch fails, there's another switch downstream or parallel to that that can counteract. So you have many backup mechanisms, or there are some control checklists. There are several checklists. Some of the switches have checklists that these are the things that must be turned on or must be okayed before going with another function. So later we'll see that. So, but again, you have backup mechanisms and other features that allow the system to operate effectively under a variety of conditions, even if you have some components that fail. And they are highly adaptable and can be modified to suit specific cell types or to respond to specific intracellular or extracellular signals. Actually, this is one of the emerging fields in biochemistry and biology in general. Basically, systems biology is one of the fields studying the systems, the control systems, the controls of the metabolic systems and metabolic functions of living organisms. Okay, so this is your cell cycle system. Rather, cell cycle control system. And the cell cycle control system, you can look at here, you have several control checkpoints. So for example, from starting with the G1, so let's start with the G1 phase here. So from the G1 is the normal day-to-day life of the cell. So when the cell want to divide, they want to multiply, basically. Basically, in biology is the only just trivia. Biology is the only field where division and multiplication means the same thing. Cell division is cell multiplication. Anyway, so when the cell wants to divide or they want to create another cell, basically multiply, of course, you must first uh, it must go to the S phase first. The number of DNA must be doubled first. So before going through with the S phase, there is a check here. Before it enters the S phase, the cell must check if the environment is favorable. So is it first of all, do we have space? We have enough nutrients and materials here. So basically, those are the environmental controls. So the first look is in environmental controls. So one of the reason why for a mature adult our cells undergo cell cycle is to replace the old and the dying cells or to heal certain wounds. And it will be sensed as an environmental stimuli. So is the environment favorable? Do we need to divide? If we don't need to divide, we won't divide. If we have to divide because we need to make up for the lost cells that have gone old or that have been injured and destroyed, basically, you need to repair it. So they must divide. So that is the reason for division. For the tumors, the reason why tumors grow, basically, tumors and neoplasms, is because this checkpoint is bypassed. Something went wrong in this checkpoint because the cells have a sense, basically, they have a sense of their environment, whether they are overcrowded or not. And tumors do not have that sense. So basically, that is one of the controls that, or rather, the conditions that must be met for the G1 to the S phase transition. Okay, moving on, S phase, G2 phase. So another important switch is here, the G2 to M transition. First of all, The question is the DNA replicated? Do we have enough number of chromosomes? And corollary to that, is the DNA replicated correctly? Or is it not correctly? Because sometimes, even though you have DNA replications, there are some mismatches there, or there might be some breaks. Is it okay? Is the DNA okay? Is the question before entering the mitosis. And again, second one is environment favorable. Another question, the same with this one. Do you have a favorable environment? So if both are yes, you enter the M phase. If both are no, go to apoptosis or senescence. So basically, that is the control of your G2 phase. Because once you have your cell entering the S and the G2 phases, it can only undergo cell division or apoptosis. Okay, because it has double the amount of the DNA, and therefore it is very unstable because it's a large amount of DNA inside. So it must either go for the M phase or die. Okay. And then once you have your M phase, are the chromosomes attached to the spindle? Is the mitosis going on smoothly? So this is during the metaphase to anaphase transition. And they trigger anaphase and they proceed to cytokinesis. So this is the third trigger here. So these are some examples. Although, take note, the controls are not limited to these three. There are many different controls during the S phases, even during the S to G2 phases, and here. But these are some of the pertinent and important ones. Okay, so what is the main players in the control of the cell cycle system? We have the Cyclin-Cdk complexes. So cyclin and CDK, these are proteins. These are messenger proteins that initiates or that is part of cascades of reactions that govern the switches between these transitions between the phases. So we have here actually four families of Cyclin-Cdk. So the first families, the first two actually, the G1 Cyclin-Cdk and the S Cyclin-Cdk complexes. So two families here. G1S. And then you have your SCDK, MCDK. So basically, these are the controls, the control switches governing these the system. So when I say they govern the system, it's not means they are the system is made up exclusively of this one. It's just that they are closely associated in RNA and basically expression studies. These are closely associated with the system. So these are the Cyclin-Cdk proteins complexes. Actually, these are one of the favorite targets for drug deliveries and drug designs, especially if you want to control cell cycles, especially in cancer treatment. So some of them try to control the CDK cycle, CDK control system. Okay, so basically, these are some examples on how it does work. For example, the G1 CDK control system, if you have a favorable extracellular environment, it will turn on the G1 CDK, turning on the G1S cyclin synthesis. And again, this will in turn activate the SCDK, which triggers the movement from the G1 phase to the S phase. So your cell will undergo DNA replication. Now, the DNA damage is a signal that stops the G1S CDK and the SCDK switch, or basically I call it a switch, but it's actually a cascade of reactions. So if the favorable extracellular environment is the go, if it pushes the CDK to move forward, the DNA damage signal stops it. So it's a negative control of that one. So if this is a positive control, it's a negative control for the transition from G1 to S phase. Next. So another one is if you have unreplicated DNA, the DNA is not completely replicated, or you have DNA damage even after synthesis. You have the MCDK, which usually governs the transition from G2 to the M. If it receives these signals, it will stop DNA or rather, it will have stop DNA re-replication. So basically, they will stop the G2 to M. they will stop basically the start of the cell division phase. It's either they repair the DNA or replicate the unreplicated DNA. So basically, that's the switch here. The MCDK stops the DNA replication and moves forward with the G2 and the M. What stops the MCDK is the unreplicated DNA and the DNA damage signal. So basically, that's that. And then the APCC complexes, which goes for the anaphase transition. What stops them is the unattached chromosome to the spindle, a signal that tells the cell you have an attached chromosome to the spindle. So it stops the ABCC. Since the ABCC is a signal for the progress of the anaphase, you have, if it stopped, basically the progress of the anaphase is stopped until the chromosome is properly attached. So basically those are the signals that you have, and these are the players, the switches that are governing for that signal. Okay, so this is for the switches, the control of the cell cycles. So let's look at the principal stages of the M phase. Basically, these are just some of the more or less a review of your mitosis. So of course, we have prophase. So you have at prophase, the chromosomes are starting to connect. So here is the actual image in fluorescence microscope of this one. So at prophase, the replicated chromosomes, each consisting of two closely associated sister chromatids, they condensed. So outside the nucleus, the mitotic spindle assembles between the two centrosomes, which have replicated or moved apart. So for simplicity, so only 3 chromosomes here are shown. So in diploid cells, there would be 2 copies of each chromosome present. So in this fluorescence micrograph here on the right, chromosomes are stained orange and the microtubules are in green. So the microtubules are the one that are guiding the chromosomes on how to move. Okay, we have the prometaphase. The prometaphase starts abruptly with the breakdown of the nuclear envelope, and chromosomes can now attach to the spindle microtubules via their kinetochores and undergo active movement. So the kinetochore is basically the one at the middle of the chromosomes. Okay, so metaphase. So in metaphase, the chromosomes are aligned at the equator. So it's actually quite visible if you look, if you have your general biology laboratory class and you did the microscopy, you will see it is very one of the more prominent things that you can actually see under the microscope. The chromosomes lining up at the middle. So they are aligned at, we call it the equator of the spindle midway between the spindle poles. So the kinetochore microtubules attach sister chromatids to the opposite poles of the spindle. So that's the metaphase. And then we have the anaphase. The anaphase, well, it's called ana because it means to separate or to cut. So at anaphase, the sister chromatids synchronously separate to form 2 daughter chromosomes, and each is pulled slowly toward the spindle pole. So the kinetochore microtubule gets shorter and the spindle poles also move apart. So both processes contribute to chromosome segregation. So as you can see here, the lined up, if it's lined up in the metaphase, in the anaphase, it separates, going to the opposite ends. And the telophase, the 2 sets of daughter chromosomes have arrived at the poles of the spindle and they condense. So you form a new nuclear envelope that assembles around each set, completing the formation of 2 nuclei and marking the end of mitosis. So the division of cytoplasm also begins with the contraction of your contractile ring. So here is your contractile ring. They are contracting to form the 2 daughter cells. So this is cytokinesis. So mitosis refers to the division of the nuclear envelope, basically the division of the chromosomes between the 2 daughter cells, whereas cytokinesis is the actual division of the cell, the cell as a whole. So during cytokinesis, the cytoplasm is divided into 2 by the contractile ring of actin and myosin filaments, which pinches the cell into 2 to create 2 daughters, each with 1 nucleus. And then you have the reformation of the interface array of microtubules nucleated around the centrosome. So that is your cell division. So in the next part of this video, we will be talking about cancer. What happens when the cell cycle controls goes haywire.